that was a very useful introduction to exciting developments coming out of the White House. And won't it be interesting to see what happens in the White House in a few months? Next, we're going to hear from Brent Stephens from Illinois Institute of Technology. And then we'll have the big discussion and everybody can get their say.
All right, thanks for the opportunity to speak here. In some ways, I'm the least qualified person in the room to give this talk, is at least how I feel, because I've only been really involved in this area for about 4 years or so, but that's given me a chance to take a look at the literature. Basically, when Katie asked me to give a talk, she said, just in 25 minutes or so, just try to summarize the entire field of indoor microbiomes, or microbiology of the built environment. So as I mentioned earlier, I'm an associate professor of architectural engineering at Illinois Institute of Technology in Chicago. Basically, that means we study energy and air quality in buildings. I first got into this field in 2012, working with Jack Gilbert on the hospital microbiome project, which I'll tell you just a little bit about. So what I'm going to try to do today is largely show the work of others that many of others have done, particularly with a focus on those that have done work in the last 10 to 15 years or so. But it'll get a little deeper than that as well. I think we'll go back about 140 years if we can. And then I will offer, so I'm going to do sort of a wide-ranging overview of everything that I think that we've learned from this. And I'm not a microbiologist, I'm not a macrobiologist, I'm not any size biologist, so I'm sort of trying my best to interpret this for a practicing building engineer and to understand what this means for building design and operation. So the introduction and motivation is pretty straightforward. We know that studying the role of the built environment and exposing humans to specific microbes, largely for many years looking at pathogens and or allergens, and also looking at the role of the microbes, how microbes are responsible for deteriorating building materials. That has a really, really rich history. This has been looked at for many, many years. And what I'm going to do with this review is propose a couple of things. And one is that the advent and then subsequent application of molecular sequencing techniques, which most of this review will focus on, has primarily increased the complexity with which we understand indoor microbiomes from an ecological perspective, physical perspective, statistical, et cetera. And as we've already heard about, it's beginning to change the way we think about health in buildings. But I will also argue that the practical implications of these advancements remains somewhat elusive from my point of view. So let's go through what I think we've learned about the built environment microbiome. And apologies to Chuck Haas and a couple of others. There's little to no water, built environment water work in here. It's a bias because I largely study indoor air. So, but I know somebody will take up that torch a little bit later. But on the left, you've got a plot of the number of citations of papers in Google Scholar, a rough search looking at microbiology or microbiome or bioaerosol and indoor. And the number of papers out there to synthesize is skyrocketing. I wish I could have given this talk 4 years ago. But we've learned a lot. So I've got this sort of list of a dozen things or so that I think we've learned. And some of these are pretty straightforward. The first one is that culture-independent methods for analyzing environmental microbiology samples reveal vastly greater microbial diversity compared to culture-based methods, right? With the advent of molecular methods, and you'll see throughout this talk, I have a lot of quotes because I haven't done a lot of work in this area. This is looking at others. The ability to sequence DNA samples from the environment has allowed scientists to detect far more than the 1% or so of the microbes that can be cultured in the lab. And so this has really led to this widespread revolution. But we've been studying microbes in buildings for a long time. I just pulled 2 examples out of the literature from about 10 or 15 years ago. And on the left, you've got samples, and these were both studies that use culture-based methods, and so, you know, an image of a culture plate here. On the left, you've got samples from 100 office buildings in the EPA-based study. So those samples were taken back in the mid-1990s. And, you know, we learned from this study. We understood how sort of quantities of microbes, and largely bacteria in these sides, on the left side here, compared indoor versus outdoor, and we started to kind of understand the types of bacteria that are found in those air samples, typically from a physical size. So we can learn, we've learned a little bit there. On the right, I've got an article from actually my predecessor at Illinois Institute of Technology, Dimitrios Moskondreos, who published with some colleagues just a study of indoor and outdoor bacteria, culture-based methods in 20 homes in Chicago. And we started to learn even back in 2003 that for the most part, you had greater amounts of viable bacteria in a bunch of indoor locations compared to outdoor locations. Indoor bacteria levels, viable bacteria levels, greater than outdoor. And they did some identification of these viable bacteria and found that a good third or so were Staphylococcus, so skin-associated or human-associated, just using these culture-based methods. And then what we started to learn, starting around 2005 or so, is that there is a vastly greater microbial diversity out there. So we're learning sort of inching along and adding complexity to what we already knew. So up top, I've just got a plot of bacterial diversity in a child care facility from a paper in 2007, Scott Kelly's group. And they found, quote, culture-independent methods based on 16S rRNA gene sequencing revealed an entirely new dimension of microbial diversity, including an estimated 190 bacterial species from 15 bacterial divisions. So we've gone from sort of what do these things look like to, you know, there's a lot more there, and particularly when you're not looking just for viable or just for culturable. A paper on the bottom from Jack's group in 2014 looked at bacterial diversity in 7 residences, and you can see the phyla largely are grouped into a handful. These are 7 different homes. On different surfaces, bedroom floor, front doorknob, and bathroom doorknob. And it's sort of a much greater mixture of proteobacteria and firmicutes and a few others. A more recent paper of looking in 2015 in the journal Microbiome looked at 29 homes in San Francisco and found really this set of diversity. So if we remember Staphylococcus, which is about a third of all the cultivated or the viable bacteria in homes in Chicago 15 years ago, only makes about 4% or so of this bit up here. And so this is a mixture of family and genus and level with their associated phyla. And so again, the point, I could go on and on, but each one of these points is going to be sort of, the title slide is what I think we've learned, and then some evidence for that. So culture-independent methods reveal vastly greater microbial diversity. The next thing we started to learn is that indoor spaces often harbor unique microbial communities. So there's a paper in 2012 here looking at bacterial communities on office surfaces in 3 U.S. cities. On the left, you've got an analysis of the bacterial diversity between these 3 locations, Tucson, San Francisco, and New York. And bacterial community diversity in the Tucson samples was clearly distinguishable from that of the New York and San Francisco samples. Why? No idea. But that's what's happening. It's a unique microbial community. And then here's a measure of abundance or number of taxonomic units on the right. And you can see San Francisco was actually lower in this measure. So there's some difference from city to city. But basically what they found, again, is that another type of analytical method found that there were more than 500 bacterial genera from 20 different bacterial divisions. And the most abundant of these tended to be common inhabitants of the human, skin, nasal, oral, and intestinal cavities. So unique microbial communities are found indoors. This is true, was also shown from a paper in 2008 looking at 2 different buildings in Finland and looking at indoor dust bacterial flora. And they found that differences between the buildings were greater than the differences within buildings between seasons. So again, indoor spaces, unique microbial communities. And again, started to hint at, well, I think most of this is coming from users of the building. Another example of this is Jack Gilbert's group's paper in 2014 in Science that looked at 7 families in their homes over 6 weeks, including 3 families that moved. And the long and short of all these is that microbial communities differed substantially among homes. There was even some ability to identify families by their indoor microbiota found on surfaces in their homes. And then, you know, some interesting things where floors resemble feet and other floors. That's good. Kitchen counters do not resemble noses, right? All right, so we're starting to learn a little bit about how these things are connected.
Genius research.
Genius research, right, yeah.
No one else had shown that before.
No. The next step, I'll switch over to fungal communities for a second, and then I'll switch back to largely bacterial, and you'll also notice viral was almost entirely absent from this until later. The sort of next big thing that we've learned is that indoor fungal communities are largely driven by outdoor fungal communities, particularly in non-damp buildings. So there's a lot of text here, but basically a paper on the left found that fungal diversity was higher in temperate zones, and literally the distance from the equator was a decent predictor of how indoor fungal communities and their indoor fungal diversity looked. And then remarkably, quote, building function has no significant effect on indoor fungal composition, despite stark contrast between architecture and materials in some of those buildings in close proximity. So fungi come from outdoors, and that's it, in non-damp buildings, for the most part. I'm trying to paint with broad brushes, and we'll miss a few things. So please help me when if someone can take a tally of all the things I've gotten wrong, that would be great. Now, the paper on the right, very similar. Fungal assemblages indoors were diverse and strongly determined by dispersal from outdoors. No fungal taxes were found as indicators of indoor air. More fungal biomass was detected outdoors than indoors. And room and occupant behavior had no detectable effect on the fungi found in indoor air. So I think that's pretty clear. However, when you get into damp buildings, it gets exciting. Once you add water to building materials, it gets exciting. What's interesting is a paper here on the left. What I argue or propose is that indoor fungal communities in damp buildings are often distinct from those in non-damp buildings. So a paper on the left, published a couple of years ago, looked at 36 flood damaged and 14 non-flooded homes in Boulder, Colorado after their 2013 flood. And you're looking at qPCR abundance of fungal abundances. And they found that fungal abundances were estimated to be about 3 times higher in flooded homes. And they were also able to determine that penicillium were the most abundant taxa. So that makes sense. What's interesting is that a paper on the right from a group of researchers in Finland, published back in 2001, did size-resolved aerosol sampling and culture-based methods in just 2 homes in Finland. And so they're plotting the size distribution of colony-forming units per cubic meter. And the dashed line is an index building that had been flooded, and then the solid line is a reference building that had not been flooded. So again, 15 years ago, using old school methods, they're in this small set of homes, they found greater viable fungi abundance in flooded homes. And they had also determined just microscopically penicillium and aspergillus were higher in the flooded building. So we're adding to this knowledge base that as an engineer goes, as a building scientist goes, there's some things we already knew. We're adding complexity to it, and we're using new tools. Okay, the 5th thing I think that we've learned, indoor bacteria are mostly indoor generated. One of the early studies here from 2008, looking at 2 shopping centers in Singapore, first of all found that looking at bacterial diversity on the y-axis, indoor air was much less diverse than environments that microbiologists and microbial ecologists typically study, soil, water, that kind of thing. But even going inside, they found that indoor air microbes are not random transients from outdoor environments, but rather originate from indoor niches. And lots of examples of this. I won't go too much deeper on that one. The 6th thing I think we've learned is that a variety of techniques for source tracking, using sort of knowledge of of skin microbiome and source types, and using knowledge of the environmental samples. Source tracking techniques demonstrate that humans and pets, so animal occupants, often dominate bacterial communities on indoor surfaces. We'll look at indoor surfaces first. So just a couple of pieces of evidence here is a paper on the left. Human skin was the primary source of bacteria across all kitchen surfaces in homes, with contributions from food and faucet water dominating in a few specific locations. And you can see the intensity or the richness of this red color has more skin-associated or palm-associated bacterial taxa on these surfaces. So that makes sense. You can sort of, identify somebody touches a trash can, you open these things a lot. I guess it's a microwave. Similar things in public restroom surfaces. Human-associated microbes are commonly found on restroom surfaces. People use the bathroom. Bacterial pathogens, also some interesting things using other techniques. Bacterial pathogens could readily be transmitted between individuals by touching the surfaces. So the big pink bars here on a bunch of different surfaces in public restrooms is the relative abundance of skin-associated taxa. And mix in with a little gut as well, which you can see down on the toilet seat. Continuing in this theme, where source tracking techniques demonstrate and really add complexity to how humans and pets often dominate bacterial communities. On the left, you've got a plot of bacterial diversity across 40 homes in North Carolina. Essentially what they found were that specific locations, even within a home, were distinct. The presence of dogs led to greater microbial bacterial diversity. And there were some correlations between indoor and outdoor communities, sort of suggesting, yes, humans and pets are a big contributor, but so is the outdoor air. And part of that was when they sampled the door trim, which is used as sort of a passive indicator of ambient air, because they assume that people don't really clean those very often. Interior looked a lot like exterior. So at least in dirty spaces that are never really clean, over time, the outdoor source of bacteria actually really contributes. And again, some really interesting, these red boxes here demonstrate, for example, pillowcases looked a whole lot like the human oral cavity, lots of drool. Toilet seats look like a lot like human skin, and soil looked like, or door trim on the exterior looked a lot like soil. Right, so they're really making some interesting connections here. Again, Jack's paper in 2014 sampled residential surfaces and found some interesting things in one particular home. You can see the black bar in each one of these plots shows when person one in the home left the house. They were gone for a couple days. And you can see if we just sort of pick one of these, person one is the dark red, sort of the signature, the microbial signature shown of person one is shown in dark red. You can see for the front doorknob, for example, after he or she left and was gone for a couple of days, their signature disappeared, and when they came back, it came back. So there's really interesting sort of identification sort of things that you can do with this. And then another piece of evidence here that source tracking techniques demonstrate human and pets dominate bacterial communities on indoor surfaces. One is some data that is yet to be published from the Hospital Microbiome project, on which I worked with Jack and his colleagues. And on the left, you've got a Shannon H index, a measure of diversity. The greater the number, the greater bacterial diversity or number of communities found in a sample. And in the blue bars, you've got pre-opening. So this was right before a hospital opened. It was built, but just construction trades and so forth were around doing finishing touches. And then red, the red plot is post-opening, so when it was occupied with staff, nurses, and patients. And for the most part, places that are highly touched, station phone, nurse station countertop, computer mouse, all increased in diversity after people showed up. But the floors didn't really change too much. But that's just one measure based on molecular techniques of looking at this data. Another measure is looking at the relative abundance of particular taxa. And you can see basically that places like the station countertop and the room floor, even though diversity and the room floor diversity didn't really change, what happened is you shifted from a taxa that's typically associated with soils, in the big blue bar here, for example, to more that are associated with human skin. So, okay, again, makes sense, right? People show up and start shedding their microbes everywhere. All right. Number 7 is that occupants, this was interesting, and there's sort of less evidence here, but it's interesting, and I wanted to include it. Occupants and surfaces interact in both directions. I've been talking a lot about occupants as a source of indoor microbes, but they also can acquire from the built environment. On the left, you've just got a plot, again, from the hospital microbiomes study of unifrac distance, where essentially, and this is looking between samples between a patient hand and the room floor. And essentially, those samples got more similar to each other in time with length of stay. So on day 0, they showed up, and their communities were a little different from what was on the floor in these rooms. But by day 3, if they stayed in the hospital for 3 days, they became more similar. And there was some evidence too that there was an asymmetry here, where basically taxa shared with the skin of the current patient are more abundant on room surfaces after they've spent a night in the room, compared to taxa shared with the room surfaces are more abundant on patient skin when a patient first enters. So it sort of suggests that patients initially pick up associated taxa that predate their stay, but then by a day or 2 later, they turn that source back around and start depositing to the environment. I haven't seen a whole lot here, but really interesting. So that's all about surfaces and largely bacteria, and a little bit about surfaces and indoor air with fungal communities. The next thing I think we've learned is that humans are also major sources of bacteria to indoor air. A couple of papers here showed in 2012, the top occupancy increased the total aerosol mass and bacterial genome concentration in indoor air, with an increase of nearly 2 orders of magnitude in the airborne bacterial genome concentration. And then if you measure a few more things in a built environment, these are measurements from a classroom. Again, size resolved, so you can sort of learn a lot more about the fate of these things. Larger particles will deposit more rapidly to surfaces and so forth. If you measured a few things like the number of people in the room, the ventilation rate in the space, the volume of the room, you could back calculate the emission rate, which is a really important parameter beyond just looking at field surveys. So essentially, these researchers were able to calculate the emission rate was on the order of about 37 million genome copy numbers per person per hour. And here was a size resolution basis. Most of them were in the kind of 3 to 5 micrometer range. So we're getting some more complexity. We already knew people emit bacteria into the indoor air. We know that. But there's complexity here that's being added that's really helpful. Some other evidence of showing humans are being major sources of bacteria to indoor air were published in 2014, again from the same Berkeley research group. This is a complicated plot, but you can just focus on the time resolved bioaerosol fluorescence data on the bottom here. So this is size resolved. And the intensity of the colors, if you've got more yellows, reds, and so forth, you've got a higher concentration of basically fluorescent bioaerosols or fluorescent particles that are in this space. So it's a measure of biological, an indicator of biological origin particles. And you can see in an unoccupied classroom, it's just a little bit of quiet, a little bit of noise. And then here's the signal when people show up, right? As you can see, you can track when they actually show up by following the CO2 concentrations in the space. So you can see occupied, reoccupied, not occupied, occupied, et cetera. And you can see these big bursts, right, where you've got, particularly in the kind of 3 to 5 micrometer range, you've got these big bursts of fluorescent biological particles. So people coming in, shedding, emitting, breathing, however they're emitting it, we'll learn that in a second, you're seeing this signal. Some similar work, or some work from the same group, continue to show this. On the left, you've got some more evidence, again, back calculating emission rates of bacteria per person, milligram per hour, and even looking at fungal emission rates. So it turns out there was a fungal signal, okay? In the bottom left here, you've got, again, the same kind of evidence. These are bioaerosol samples in a few different cities. And you're looking at the indoor-outdoor ratio of bacteria. Occupied classrooms are the black dots, and then the white dots are unoccupied. So basically, when it's unoccupied, you're around indoor-outdoor ratio of airborne bacteria of about 1. Indoors equals outdoors. And then when people show up, you get huge increases, right? Fungi, you get some increases, but not as much. On the right, you've got an interesting plot where they took their samples, and because they had measures of ventilation rates, they could back calculate the relative contribution of ventilation, meaning outdoor air brings in the microbes, versus emissions, meaning people emit them in any form. And the black bars are emissions. So in most of these different cities, classrooms in these different cities around the world, indoor emissions dominated bacterial concentrations. But there were a few where ventilation was an important source. So we're starting to add complexity to, okay, we know people emit indoors, but we also know bacteria can come in from outdoors. There's some balance here. Fungi was actually not quite as stark as what we had seen on surfaces, right? I originally said fungi only come from outdoors, right? In non-damp buildings. But here you've got a mixture of about half came from indoor emissions and about half came from ventilation. So what are those emissions? Well, it turns out you've got to do some sort of more controlled studies. And we sort of finally got to these controlled environment studies around 2014 or so. And so now, what I argue is that controlled studies can elucidate the mechanisms of human microbial emissions. And the mechanisms are threefold that I'm aware of. One is direct shedding, coming directly off of us. The other one is resuspension of settled particles, so walking on carpets on which particles have deposited, resuspending those. And another is just direct surface contact. Every time I hit this podium, there's some transfer between me and the podium. In which direction, I do not know. And so these controlled environment studies can really elucidate these things. And so just a summary, again, of some work, this is in 2016 from the Berkeley Microbial Ecology Research, Indoor Microbial Ecology Research Consortium, found a few key points here that I think are worth looking at. And again, this is again using the UV aerodynamic particle sizer. So it's just looking at real-time fluorescent, bioaerosol fluorescence. And they found that seated, simulated office work conditions, pretty mild work conditions, generated about 1 million particles per hour per person using this instrument. Getting up and walking around increased this about 5 to 6 times. And they found that was actually mostly attributable not to direct shedding, but to resuspension. There's some arm movements, as sort of fibers and other things are coming off of my suit. There was some evidence that was contributed to the increase. But really, during both walking and even sitting, where people shuffle their feet a little bit here and there, more than 2/3 of the emissions originated from the floor, is what they back calculated. So resuspension was dominant. So again, we're learning more about how and why people emit bioaerosol particles, in this case, into the indoor air, which then can go on and exfiltrate, be exhausted out, or deposit onto surfaces, or land on our mucous membranes and other things. And again, the dominant particle size is 3 to 5 micrometers. I only have 11 things, so I'm at 10. The next thing that I've sort of noticed in the literature is that building design and operation can influence indoor microbial communities. And a lot of this work came out of Jessica Green's group in 2012, where on the left you've got a plot of phylogenetic diversity with 3 different ventilation conditions in a healthcare facility. On the right you've got outdoor air, so the most diversity there, and then 2 different ventilation conditions, sorry. On the left, you've got indoor mechanical, and in the middle, you've got indoor window. So essentially, when you open the window and naturally ventilated this space, which those of you that aren't building scientists, natural ventilation just largely means open windows or flow through holes in a building. You had greater diversity, and essentially, on the right here, indoor air became the microbial communities in indoor air started to look more like outdoor air. And then when you shut the window and operated and delivered ventilation through the mechanical ventilation system, that diversity decreased. So we had a lot lower diversity depending on how we operated the building. I say we, how Jessica and her team operated the building. And essentially, again, more evidence that bacterial diversity was lower indoor air than outdoor air in both scenarios. There was also some correlation with temperature and relative humidity, but they're sort of correlated with the source of ventilation air, so I'll talk about that in a second. This is just about building design and operation. Again, a couple of years later, her group looked at bacterial communities in classrooms and in offices, and again found the same sort of thing. Window versus mechanical ventilation changed the types of communities that were found indoors. Within offices, quote, the source of ventilation air had the greatest effect on bacterial community structure. And a couple of plots up here, even if absolute abundance stayed the same, the relative abundance of different taxa changed depending on where the outdoor air was coming in from. Just a couple of examples from there. Some more work from her group also found sort of an interesting architectural measure. So building design and use, really, was also associated with differences in bacterial communities. And this was the degree of connectedness of spaces, just sort of a geometric and occupant-based metric of how many people are moving through a space because of how it's connected, right? So you could have these different shapes where this isn't very connected, but this space is very connected. You've got lots of people coming in and out of there. And they found some measure of biological similarity between the communities that they were looking at and this connectedness distance. So basically, when places were more connected to each other, their microbial communities looked more similar to each other. So interesting. And then finally, to get back to this sort of balance between indoor emissions and ventilation as a source, to further show that building design and operation can influence microbial communities. Again, on the left is a plot from Jessica Green's group, and on the right from the Berkeley research group, that both showed that, well, when you actually then really increase ventilation rates in buildings, even if it was using mechanical, if you brought in more outdoor air, you actually had the indoor air communities more closely tracking those of outdoor air. But we know that we knew that from other physical and chemical studies for many years. This is just showing it again from a biological perspective. And then the last point I want to make before offering some thoughts on all this is that building conditions in general have appeared to have a small influence on indoor microbial communities. Building conditions, I generally mean things, built environment parameters, temperature, relative humidity, that sort of thing. So here's a big plot to confuse you of long-term environmental conditions measured in a new hospital that we were part of with Jack's group. And again, this was the same hospital that we measured a couple days or a couple months before it opened, and then for a year after it opened. And we just had widely varying environmental conditions: temperature, relative humidity, absolute humidity, even light over here on the far right. We had widely varying differences in occupancy, the rate at which people came in and out of these single occupancy patient rooms. And at the end of the day, again, this paper's in review right now, we saw really minor signals, correlations between the bacterial community analysis, largely 16S, relative abundance data. We did see that higher temperatures and higher luminance were associated with greater microbial dissimilarity between patient and surface microbial communities. So basically, higher temperature, higher luminance, that means the communities on patients, on their skin, and surfaces nearby them were more different. Physically, mechanistically, I don't think we have a really good explanation. It may just be a trigger, a signal in the noise. Or a noise within the noise. But the impacts are really small, even if we do see them, okay? And then really, I show this because it was one of the first studies that we threw a bunch of building environment parameter measurements alongside a microbial assessment. A similar, or another study was going on around the same time from University of Toronto, that led to this paper and editorial that was just published about 2 months ago. They were looking at microbial communities on surfaces in conjunction with different environmental and built environmental parameter that they were measuring. And they basically led the editorial to say the built environment is a microbial wasteland. Because essentially what they found was there was no correlation with any of the built environment parameters that they had measured, where they had characterized different kinds of surfaces on the floor, on the wall, and on the ceiling. They basically found there was greater diversity, i.e. more different types of communities on the floor than on the wall and ceiling, which from a physical perspective makes sense. And they found no correlation with bacterial or fungal community diversity with moisture parameters that you would think would have a really big influence. They really didn't find anything. Basically, the long and short was this stuff deposits, and that's it. You know, not active, it's not growing. There's nothing amenable in most surfaces for these things to be growing. They're dormant. These aren't viable measures. So we're just detecting DNA, and that's largely it. And then so there's that. But then you can also look in the literature and find a little bit of different evidence that building conditions can often have a small influence on indoor microbial communities, but sometimes a little bit larger. So it's not clear sort of where this is going. The first was finding some correlations of the proportion of bacterial sequences closely related to pathogens in one of Jessica Green's studies. Was correlated with relative humidity and temperature, but it was also, those factors were correlated to some extent with how the room was operated and where the samples were coming from. There's a little bit of evidence from Rachel Adams' paper in 2014 that found bacterial richness tended to be higher in 4 of the 11 residential units that they had measured in that reported at least occasional humidifier use. Well, again, it gets interesting when you bring water into buildings again. And then just a couple of other papers there. Karen Dannemiller's recent paper in Indoor Air looked specifically at the influence of housing characteristics on bacterial and fungal communities in homes of asthmatic children, and basically found that increased microbial richness was associated with the presence of pets, water leaks, again, longer air conditioning use, even their locations, suburban versus urban homes, as well as some other dust composition measures. They also found that occupant density measures were associated with some beneficial bacterial taxa, including lactobacillus. So there's some evidence there. Part of it has to do with the study designs and the outcomes that they're measuring. And then the last just piece of evidence is a study from 2016, just published about a month ago, looked at fungal and bacterial communities and indoor dust, alongside different environmental determinants. So they looked at house dust, fungal communities and house dust in almost 300 homes. And they found that the biggest predictors of fungal diversity was surrounding greenness. How many plants, what kind of plant coverage is outside? Again, indoor fungi coming from outdoor. Also, some outdoor particulate matter concentrations. The age of the building, that might have to do with its air tightness and its ability for outdoor air to infiltrate. And of course, window opening behavior. So these may all be surrogates of the same thing. You know, more foliage outdoors and greater amounts of outdoor air ventilation. So that's sort of where we are from an architectural engineering standpoint. So I just, mostly for the study committee, I just list all 11 of these in case you want to use them. This is a summary of some of what we learned. And if I've grossly oversimplified some things, I apologize. But this is from a practical sort of standpoint. And then I didn't know how to show this, but I really wanted to show this paper to understand what we've learned and where we're going from a practical standpoint. Has anyone read this paper? Hal? It was brought to my attention. It's sort of delicate how you talk about this, I think, but it's really fascinating. Mostly, it's just one of the most fascinating papers I've ever read. This was brought to my attention in a review paper a couple years ago by James Scott at the University of Toronto, who said, you know, we've really learned a lot, but we also learned a lot a long time ago. So this is a paper from 1887. I think Scottish researchers, I believe. The title is The Carbonic Acid, Organic Matter, and Microorganisms in Air, More Especially of Dwellings in Schools. I've got to start rethinking the way we title our papers. So basically CO2, which we measure in buildings all the time. Somehow we've made a living out of doing it. Andy too. Organic matter, so some measure of organics and microorganisms as defined and measured at the time in dwellings and schools. And this paper is fascinating. So one of the I have a lot of excerpts from this. This is an investigation into the sources of organic matter and microorganisms of air inside buildings and the circumstances affecting the number of microorganisms, also the relative number of bacteria and molds in both outside and inside air. And how did they measure? They did air sampling, 1887. They took this sort of glass tube with a cloth over the top and lined it with meat jelly and ran air through it and collected microbes on this thing and then looked at them under a microscope or however powerful that was at the time. And so we learned some things that from a study like this that go beyond just microbiology, the built environment. So this is, adding to the evidence is what we've been doing since 1887. The first thing they mention is key for any indoor air quality investigation. Always compare indoor air to outdoor air. In order to draw conclusions from an examination of air inside buildings, it is of course necessary to know the state of the outside air. As regards each of the constituents estimated, considerable variations were found at different times and places. They found that occupant density drives bacterial counts but has no impact on fungi. Interesting. So these are cubic space per person. They basically went into a bunch of different kinds of homes. They categorized them by how wealthy they were and this sort of thing, or sort of there's some real classes stuff in here that's a little odd, but. Basically, on the high end of this, you've got a small number of cubic feet per person, highly densely packed places, and then the rich lived in these bottom areas where they had large, 4,000 cubic feet per person. And if you look over in the right-hand column, bacteria was highest in the densely occupied spaces and lowest in the less densely occupied spaces. Molds or fungi didn't really have any impact. Oh, all right. I think we knew that. The explanation of the ratio bacteria to molds increasing with the vitiation of the air of the molds come mostly from the outside air. Fungi come from outdoors. That's great. When the air in a room becomes vitiated, the bacteria increase largely, while the number of molds is affected to a relatively much less extent, if at all. Another one, we learned about resuspension in 1887. They wanted to know where are these, if it's coming from people, how's it getting into the air? It has been shown that when a room is left quiet, the microorganisms settle out in a few hours so that the air becomes comparatively free. Hence, it is clear that a certain amount of physical disturbance in a room is a condition necessary to the presence of microorganisms in air. It might naturally be supposed that the effects of physical disturbance would tend to obscure all other factors. A few other quotes here. Be expected that a large number of bacteria would be given off from bedclothes when shaken. I've seen some studies on linens lately. Also, the skin and clothes of the persons present in the room at the time of an observation also naturally occur as a probable source of infection of air, right? Infection of air. Bacteria getting to there, it's infection. That's certainly changed. This source, however, is much less important than might be supposed, say, we think, be shown from our observations. So skin and clothes of persons. Then they went on to sort of show that the pig pen effect is very real. They made some measure of the cleanliness of rooms and the persons in them. So the subjective nature of this could be in question, but they basically characterize people and their homes as clean, dirty, dirtier, or very dirty. And they found a correlation with microorganisms. More microorganisms were found in the dirtier homes with the people who were apparently dirtier. So again, resuspension and sort of this cleanliness nature. Again, a couple more from this, and then I'll wrap up. This source and rate of ventilation air delivery impacts indoor microbes. So they had two different types of spaces here. One was mechanically ventilated, which actually brought in more outdoor air than what they call naturally ventilated, which is really infiltration, not open windows. It's just closed windows. And they found through and through you had greater bacterial counts, 9-to-1 ratio, and then in the unventilated buildings rather than the mechanically ventilated buildings. Molds were a smaller impact, or fungi were a smaller impact. And I love this. We're still, arguing for better ventilation in classrooms from an indoor air quality standpoint, so including a wide range of physics and chemistry and biology that influences that. The all-important argument for mechanical ventilation is that it maintains a certain standard of purity, and unless some simpler method which will maintain a similar standard will be devised, its adoption in crowded schools seems to be very much required. Anyone that was at the Sloan Boulder conference that a couple of weeks ago that showed ventilation rates in classrooms and outdoor air intakes being shut up and all that, we knew in 1887 that you should ventilate classrooms. And then the last piece that I want to show from this is that indoor air microbes, or indoor microbes and built environment factors are actually weakly correlated. They measured CO2 or a measure of it, and these microorganisms, they ain't finding a correlation. Same thing in the hospital study. So my sort of, what I see here is that to summarize what we've learned from 1887 to 2016 and moving forward, is that there's a quote from this 2007 paper that I cited very early on from Scott Kelly's group that I think we've kind of gotten away from a little bit, but I think it's really important. And it's that the combination of culture and culture-independent methods provide powerful means for determining both viability and diversity of bacteria, in this case, childcare facilities, but I think that's in all indoor environments. So we at this time in 2007 have little comprehension of the true extent of microbial diversity, but to really get there, you want to combine some of these methods, which we don't see a whole lot, so you can understand viability and so forth. So I think I'm going a little bit longer than I should have, but I've got another maybe 5 minutes or so. So where are we going? There's two things that I think we see immediately. One is that we continue to build upon the existing knowledge primarily by adding complexity. So physically, ecologically, statistically, we add complexity to this. And we're also beginning to think different about microbes in indoor environments and health, but we're kind of stuck in between two positions. So I want to show share two thoughts on this. The first comes from the complexity of sneezes. So Nature had a highlight on a professor, a mathematician at MIT, a couple of weeks ago that I saw on Twitter because Paula from the Sloan Foundation had tweeted it out. And I had these great visualizations on the fluid mechanics of sneezes. This is a sneeze captured on a high-speed video where large droplets of saliva and mucus, that's green, shoot out of the mouth but fall relatively quickly. And then a turbulent cloud carries the smaller droplets, the red, and allows them to drift up to 8 meters or so. And there's really great videos of all these high-speed videos of these sneezes that this group is working on, analyzing the fundamental fluids of what's going on here. And I thought, I've seen this before. I've seen super slow-mo videos of sneezes. I saw them on Mythbusters in 2010. Right? Now, of course, they weren't analyzing the fundamental fluids going on, but they were trying to see, what does this look like? I don't know why he's mashing his head into a tennis ball, but essentially you see the large droplets falling, and you see the smaller droplets, or what end up we call droplet nuclei. They look like they disappear, but they don't, right? They don't really. You expel, if you've got some respiratory pathogen and infection, you may expel individual virus particles or whatever you may be infected with at the time. And they basically just become small enough that you can't see them anymore. So they get below our visible detection. So here's one zoomed out a little bit more where he's drinking some Kool-Aid or something. And I think these are feet measures. You can see it looks like it disappears, but it doesn't. The matter doesn't disappear. It's just invisible, right? I thought surely Mythbusters wasn't the first person to look at sneezes, right? 1939. The infection of air, bacteriologic and epidemiologic factors, Wells et al. And these are just crude sort of, initial looks at the fluids of sneezes and with different kind of instrumentation. So it turns out we know a lot about sneezes, and we've learned a lot about sneezes since 1939 and before. We know that when a person coughs, speaks, sneezes, or even breathes, that particles of liquid water and proteins and other matter, salts and so forth, are expelled. These are droplets. They quickly evaporate once they leave the humid environment of our bodies, and they evaporate into these smaller droplets, right? Typically half the size of what they started with. We've learned about airborne transmission of diseases, right, for a long time. And I want to show an example of my favorite experiment of all time, if anyone's familiar with this. This is aerosol transmission of rhinovirus colds, published in 1987. I just show the text here, but basically the methods are fantastic. 27 men were inoculated intranasally with a rhinovirus and go home for a couple days, come back when you feel terrible. They came back and said, all right, great. Now we're going to split you into two groups. One group, you're going to play, in both groups, you're going to play stud and draw poker for the next 12 hours. And then in one of those groups, you were free to move as you needed. So you could touch your cards, touch your face. You're all in a circle together, but you could, all modes are active. Surface transmission, fomite transmission, direct and long range aerosol transmission. In the other half, you're going to be harnessed in this brace where you can't touch your face. You can only deal your cards, right? You can only deal your cards. So you eliminate the potential for going from a mucous membrane to the card and passing on. And what they found is there's no statistically different rate of infection in the two groups, which means that airborne was really the key component there. So just one of my favorite studies. An anecdote there is that if anyone knows David Mills, who's a microbiologist at UC Davis, I was telling him about this at a Sloan Boulder meeting once, and he said, "Wait, was that University of Wisconsin?" I said, "Yeah, it was." He said, "I was a volunteer in that study." Yeah, well, he got paid, but yeah, so still just amazing. Okay, so anyway, moving on from there, we've added complexity to this. So 10 years later, people started having people cough into boxes and measure the particle size distribution. So how big are these particles? Because their size really determines whether they float around the room or deposit on surfaces. And you started finding that almost all particles that we emit in coughs are less than a micrometer. Well, then fast forward another 10, 15 years later, and we started detecting viral, influenza RNA using qPCR in aerosol samplers. First in which people coughed in, and then later just found around typical indoor environments like healthcare facilities and daycares, that kind of thing. So influenza viral RNA floating around, right? And actually found that although 90% of the particles that we emit are less than a micrometer, about 40% of all the viral RNA is in that fraction. You would think that the number of organisms sort of scales with the volume of the droplets that are emitted and so forth. And then again, to come back to this, that's nice, that's interesting to know viral RNA is there, but will that lead to, infection and so forth? So then you've got to kind of combine, this is just some data of influenza survival on surfaces in a 24-hour period. You've got to combine sort of, you may be able to detect the, in this case, genome copy. You may be able to use molecular methods to detect the DNA or RNA, but you also want to know, is it still viable, right? So same sort of experiments. You detected the RNA, but some other measure of an assay for its viability decays a lot faster, right? So there's this element of even particularly in the infectious disease world, it's important to kind of combine both of these biological methods as you can. So basically, what are we doing? The complexity of sneezes. When we go back to that video that was featured in the Nature article, they're not just showing that this is what it looks like. They're understanding the fundamental fluid mechanics, and they find that this sort of the physical mechanisms of how this forms. There's this breakup of fluid that occurs outside the respiratory tract. There's like these sheets that are formed that then lead to what they call tag bursts, to what they call ligaments, which finally break into droplets. So they're adding complexity to something that we already really know, right? And that's science. That's how it works, right? So, but from a practicing engineer, from a building engineer, it's not clear, how much more helpful that is. But it's certainly interesting. It's certainly worth supporting. And then the last thing I have is we've got this we're sort of moving from pathogens, thinking about pathogens, to thinking about protection, right? And Elizabeth, you mentioned this briefly earlier. There's this long history of infectious disease and asthma and allergy research that has primed us to consider negative health impacts. When you talk to engineers, they all think about microorganisms as bugs and infection-causing bugs, right? And to some extent, the general public is there too. But on the opposite end, you've got some measures of microbial diversity or abundance have been associated with positive health effects. So one of Susan Lynch's studies I cite here, and one of Karen Dannemiller's studies I cite here, where basically, there's beginning to see this evidence even in built environmental samples and house dust and so forth, that the right number of the right kind of microbes can be beneficial for health. And so I was delighted to hear that someone from Sherwin-Williams was actually here, because I have a picture of some paint on this slide from them. And so how will this all change the way that we design and operate buildings? On the left, you've got the way that engineers and many citizens have been thinking for many years, which is antibiotic. It's keeping the wrong microbes out. Just last year, an article from USA Today here, a press release, new Sherwin-Williams paint kills infection-causing bacteria. So paint shield, paint it all over the wall and kill the bacteria. And then on the right, you've got Jessica Green with this editorial Indoor Air a couple of years ago, Can Bio-Informed Design Promote Healthy Indoor Ecosystems? And I've also got a picture of a product from a company that sells these sort of probiotic juices that you can connect right to the HVAC system and deliver into the space. And so that's the probiotic, the right microbes at the right time. And we're in the middle of this spectrum somewhere. And I think there's a combination of both that you want to achieve. And I suppose that's what I would say that's really a big part of where I see this field going in the next 10 years or so. So thanks. Sorry if I went over a little bit on my time. If this field wasn't so full of work, it would have been a little shorter. So thanks, and I'll ask any questions, take any questions.